Okay, so let's talk about the genetic architecture of complex traits. And basically, one school of thought tells us that the genetic architecture of complex traits is additive. We have many variants in the genome, but each of which contributes independently to the predisposition of the trait. On the other hand, we have another school of thought that tells us that there is pervasive epistasis. And so basically the interaction between these variants, this is what's really important for the predisposition of a complex trait. And there is long-lasting debate in the field, goes to days of Fisher and Wright, what is the architecture of different traits. And of course, it doesn't mean that there is one answer. It can be that for different traits, we might have different answers depending on the trait. If the additive school of thought is correct, when I look at the correlation or the covariance of a complex trait, basically I will see that the covariance structure should follow linearly with the genetic similarity of individuals. So basically, if I look at the genetic similarity, going, let's say, from second cousins to identical twins, without the effects of the environment, I should see linear increase in the correlation of the trait. If epistasis is the right model, instead of looking at this additive equation, we should see a polynomial. And the degree of this polynomial n is the number of interacting genes. N equals 2, 2 genes interact together, n equals 3, 3 genes interact together, and so on. I want to stress the point that the coefficients of this polynomial are non-negative numbers because they represent a fraction of variance, and variance by mathematical definition is a non-negative number. So instead of looking at a linear trend, we should see a convex trend. As we go to identical twins, we should see a more rapid increase in the correlation structure. Now, in genetics, in the last 70 years or so, we did a fantastic job in looking at the correlation structure by analyzing monozygotic and dizygotic twins. But if you really want to differentiate between these two different models, we need to go to the Wild West. We need to go and start to ascertain second cousins, first cousins, and these distant relationships to really differentiate between these two trends, two different curves. So what we need is a large number of large pedigrees. But it's really hard to get a large number of large pedigrees. I have a small lab at the New York Genome Center. We cannot get suddenly millions of people by just putting ads on the New York subway and collecting these second cousins. So instead, we decided to take a very different approach and to use crowdsourcing using social media. So we focus on this website called Genie.com. In this website, genealogists can upload their family trees to the website. And if they upload, let's say I upload my tree, and let's say Jonathan uploads his tree to the website, if there is a match between the two trees, the website will offer us to merge the two trees together, and we can also collaboratively work on these trees. So this way we can construct a family tree that is beyond the limitation of knowledge of each one of us. And what you see over here is my own family tree in Genie. So with permission of our IRB and Genie.com, we downloaded all the public information from the website. We started this study in 2011, and we downloaded the first round, and then in 2015 we did another round of download, and so far we collected 86 million public profiles from the website. And in the last round, it took us 3 months to download the data, another 6 months just to organize the data into data structures that are amenable for scientific analysis. In some very rare cases, but they were there, we found people with more than 2 parents, cycles in the tree. This is not consanguinity. We expect directed acyclic graph, but we found cycles. We had to clean all these things and to get to this, and really to put the data in the right data structure. So the question is, can we get to these large pedigrees? And the answer is yes. Here is a pedigree of 6,000 individuals that spans about 7 generations. Every green node is an individual. Every red node is marriage. But this pedigree is, in fact, one of the smallest pedigrees we have from this data set. Our largest pedigree has 13 million people that are all connected through marriages and shared ancestry. So I really wanted to draw this 30 million people pedigree on this screen, but I realized that the resolution of the screen has about 1 million pixels, so it's a bit of a challenge. So as a compromise, we're just going to visualize 70,000 individuals, or 0.5% of the data. So if you look at this computer screen, think about that. We had to multiply it by 200 times, and then we could visualize this entire 30 million pedigree. And you see these massive clusters of individuals that form these like second, third, fifth, and so on cousinships in our data. All of them come from the same one big network. We have some famous people in this pedigree. We have Cyril Wright as a research subject. And we also have Kevin Bacon. And the Bacon number of Cyril Wright is 24 steps through shared ancestry and marriages. Okay, now I don't want to stand here and to say, this 30 million pedigree is all correct. Christine told us this morning about, challenges of genealogists to really document their data. So to overcome this challenge, we decided to do genetic tests. So these genealogists also put their data in other websites that we can download, and we could focus on mitochondria for subset of individuals. We had mitochondrial data and also Y chromosome data. And this spans hundreds and thousands of meiosis events that we could compare. When we look at the mitochondria and we look for mismatches between individuals, we found that the error rate, the likely error rate per meiosis is on the order of 0.3%. It's great. For the Y chromosome, the non-paternity rate was 2%. And we were really excited to see this error rate in our data, because if you look at the literature, you find this is a meta-analysis of 67 previous reports of non-paternity rates. And in Europeans, 2% is basically what this study was reported before. And there is a more recent study by Jobling found a similar figure of about 2% non-paternity. So although our data came from social media, from this collaborative work of many genealogists, what we get is something that is in similar qualities to what you expect if you ascertain these pedigrees using traditional methods. Okay, if you want to do genetics, you need to know something about the environment. For millions of these individuals, we had the exact birth location and death locations because the genealogists documented these locations as free text. Using some geoparser tools, I will not go over the pipeline, we could actually convert this collection into longitude and latitude and really to pinpoint each profile. And then we took all this data and we just drew it on a black rectangle, and this is what we found. We got a map of the Western world. Each individual in this figure, each pixel in this figure is an individual in our data. And you see we have we see a very good sampling from the Western world, from North America, from West Europe, and also a bit from Israel. Makes me very happy about it. Now, we thought my student was very excited about this graph, and she thought, what would happen if we stratify our data based on the year of birth of individuals and just visualize the data? We just create a time lapse of visualizing the data with correlation with non-historical migration events. So she created this movie. So our deep pedigrees go to the UK and Western Europe. And then Columbus arrived to America, and you start to see some profiles over there. The arrival of Mayflower. The Dutch settlement. It's a bit harder to see this one. Australia, the Cook voyage, and then everyone is going to Stanford. So this is part of the fun things that you can do when you have a large data set. And in fact, we have just another study about migration patterns and how populations disperse with this data. I'm not going to talk about it today. So let's talk about the complex traits. We decided to focus on longevity. Longevity is a complex trait, and for several reasons we focus on longevity. First, it's very easy to get longevity readouts from our data because we have over 1 million individuals with exact birth of dates and date of death in our data. Also, longevity is a complex trait, and previous GWAS did a quite, underwhelming job in finding the genetic basis of longevity. And it's kind of like a super phenotype because many diseases contribute to longevity: cancer, heart disease, diabetes, and so on. Just to convince you that the data is quite good for longevity, here is the average lifespan of our data. You can see the effect of the Civil War, of the First World War, and Spanish flu, and the Second World War in our data set. And if we take this data and look at another data set published in Science over a decade ago using vital records, you see a very good correlation between these two data sets. So although the data was collected by, again, social media, genealogists that are just interested about their family, we get quite good correlation between these two data sets. So let's try also, because we need to tread lightly with this data set, just to replicate previous results about heritability estimates in longevity. The way that we define longevity is that we take the place of birth of the individual, the year of birth, and we subtract these fixed effects from the data. We build a model, we look what is the estimated longevity, and then the longevity readout that we're going to analyze today are just the excess of longevity or the depletion of longevity from what is expected for this individual. So then we can take and have a longevity readout for both parents, average these two individuals, and just regress the mid-parent value on the offspring, and this regression tells us what is the heritability of longevity. And what we got basically, it's not the right, yeah, sorry. So we got something in the order of 17% heritability from this process, and this is well within the heritability estimates of twin studies, which is between 10 to 25%. Again, we could replicate previous studies with this data set. So now we are ready to compare between these two models. So we took our data, and here we had 3 million data points of pairwise individuals, basically pairs of relatives in our data. Here is the genetic similarity in beans of 5% versus the longevity correlation, and we fit 3 nested models: additivity, epistasis of 2 genes, and epistasis of 3 genes. And then we fit the 3 models. We look at the ANOVA results of the 3 models, and additivity was the best results. We didn't see any significant value for the epistatic models. We also tried to use BIC to differentiate between these 3 models, and BIC also favored the additive model. Last, we also did a leave one out approach. We took one class of relatives, removed it from the data, feed the 3 models, put back this class, and calculated the error of all the individuals in the class to the models. And again, additivity was the most favorable model. But we are skeptic geneticists. We wanted to have more evidence for the additive model. So if you think about it, we removed all the monozygotic. We didn't have really monozygotic twins in our data because every time we saw 2 brothers that were born the same year, we just removed them from the data set. Yet we could take these 3 models and extrapolate them and see and predict how monozygotic twins should be correlated. Then we contacted the Danish Twin Registry and obtained 900 monozygotic twins. We never used this data before. We put these monozygotic twins, the correlation that we identified in them, and you see how perfectly the additive model predicts these monozygotic twins. We wanted to do more tests. We thought about, what about the household environment? You know, maybe the household, somehow we didn't model that correct in our data. So to correct for the household environment, we also removed the effect. We look at the spouses of individuals. And spouses, if they're genetically unrelated, then they're really good surrogate for the household effect on the longevity of an individual, so we just regress it out from the longevity readouts we had, and we repeated this analysis. So we did also regression, we regress out the spouse's longevity. We considered also dominance in our model. We repeated this entire analysis just by taking lines that are from females, because the data is more, the tree is more accurate for females. All of them, I didn't put the raw data, but all of them had the similar conclusion. Additivity was the best model that described the data. We don't see epistasis in longevity. So, additive genetic architecture. Now, the data is totally available for you for download in familylinks.org. You can download data, it's de-identified, and you can play with it for your own studies. Okay, so far I told you about how we can create these big pedigrees. But we don't really have DNA in these pedigrees beyond this like tiny bit of Y chromosome in mitochondria. It would be really cool if you could add DNA. So we decided to do another project. And we thought, you know, instead of just really genotyping people, because we don't have funds for that, can we just crowdsource the DNA from individuals? And right now, we have wonderful companies like Ancestry DNA and 23andMe and Family Tree DNA that test individuals and test millions of individuals in their databases. And these individuals can really download the data. And if you think about the future, this trend is only going to increase the number of genomes that we will have available for individuals in the general public. In fact, in the future, probably everyone is going to get sequenced. So why not to put a website where we can now actually crowdsource back these data sets and from the .com to the .edu and get it back. So if you put a website, you know, the mantra, if you build it, they will come. So we put the website, we spent some time on it, and we crossed our fingers, and they came. And we have now, we put the website about 4 or 5 months ago, and we have now close to 15,000 genomes in this website, people really come and upload their data to our website. So the question is, why do they come? And there is now a lot of discussions in the Precision Medicine Initiative how to convert research subjects to research participants. So what makes people really to come and give you the very intimate data? So here are some things that, at least in our, I think, worked for us, and lessons that we learned from this study. The first thing, they come because we reciprocate. We don't tell them, people are really sympathetic to this idea that to contribute their data to science. But all of us are super busy, and we have so many things going on that really to take the time to give your data to science, and it takes some time. Most people will not bother doing that. But in DNA Land, we try to immediately reciprocate. You upload your data to our website, we give you something back. What do we give you? We give you an ancestry analysis. We take your data, we impute it using the 1,000 genomes panel, and you can download your imputed data. You're basically like as if it was whole genome sequencing for the common variants. And also, we will search relatives in our data set. And you might ask, like, okay, but the companies also search relatives in their data set, so what's the big deal? The nice thing about our data that if you were tested with 23andMe, you can now find your relatives from ancestry DNA. So you can really now break these silos and find people that you didn't know that are related to you from one database that you used. And this is our relative finder report, just a portion of it. We also allow individuals, by just one click using the Genie API, to connect their Genie profile to our website. So now basically, if they see a match, it's not just see the email address and have to start this laborious process to understand how they are related to people. They can just click on the Genie profile, go to the tree, and without the other person's participation can start already tracing these genealogical connections. Most of our participants are really into genealogy. This is why they come to the website. Another reason why they come, because our consent is simple. It's 1,500 words and written not for Harvard JDs, but with plain English that people can understand. It's animated. We have this icon, so it's very easy to understand. It's balanced. We don't promise privacy. We said we cannot really promise guarantee your genetic privacy, but we have some skin in the game. So both Joe Pickrell and I, that is the co-PI on this project, we put links to our genomes as part of this consent. So we cannot guarantee your privacy, but if something happens, you can already, it's going to affect us as well. And we're really happy to see the response of the community. So Misha Angry said that he doesn't need to take a shower after reading our consent. I think it's a good sign for the consent is working well. And a third reason why they come is because we need to create a dialogue with this community. And in fact, the 2 weeks, I was not prepared for that. We put this website, we got hundreds of people in the first few days, and we got many questions from people. So my inbox was just filled with questions of participants about how we use the data and why the website doesn't work for this sort of file. And here are some examples of individuals. Some of them have great suggestions, by the way. We really improved our website by listening to these people. But after a while, I just realized that I need to find someone. So we have Richard that joined my group. He comes every day. Basically, he's our help desk team of one person just to interact with these people, make sure to address their answers. So if you do something kind of like grassroots effort to collect scientific data, this is a lesson that we learned the hard way. You need to put someone to actually interact with them and to reply emails of people. These are not just research subjects that take the data and go away. Okay, so last thing is how we're going to collect health information. We think questionnaires are great, but I think the future is to move away from questionnaires because they take some time. In the beginning, there is some enthusiasm, but questionnaires tell you something about the health of an individual in a specific time point. And you can only ask people to answer this amount of questionnaires. So our hope, what we're planning to do in the next round of features in the website is to introduce the ability of individuals to contribute their social media data, basically their Facebook data or their Twitter data. And there are some studies out there that show that you can actually get, at least for personality traits, you can really get someone's data and you get something that is in similar quality as if this person took a thorough standard personality test. So this could be one way we're going to test it if we could really recover phenotypes this way. If we can also convince our participants to donate this type of data. And of course, there is another study here in Stanford about using searches to collect some pharmacogenomics data. So I think it's something quite promising. So I just told you today about how we can build a population scale family tree, starting to add a DNA layer to this family tree and our plan for health information layer. And I think this is something that could be quite exciting, especially with efforts as the personalized medicine initiative, how to really reach to very large data sets without investing tons of funds and efforts to regenerate these data sets from scratch. And these are the great group of people that work on this award and my funding. And thank you very much. Ready to take any questions.
Thanks, Yaniv. I was wondering, when you calculated heritability using Gini data, did you stratify by era, for example? Like,
yeah.
Okay.
Yeah, I didn't show this graph. We stratify by era. We don't see any trend. We went to 1600 all the way to 1950, and we stratify basically on parents that were born these years. And we couldn't see any, so it fluctuates, but it doesn't create any trend that is detectable by the data, that the heritability is, let's say, higher recently versus in more ancient data.
So it was
strata.
Like around 10 to 20, you said?
We took every 25 years. And okay. I can show you the slide later on. I have it. Yeah.
Okay, maybe later. Thank you.
Yes.
I was wondering if you could go to the slide that shows the plot of relatedness against heritability for longevity.
Mm-hmm. Can we get the slides back? Thank you. Let me just do this.
So while you're finding it, I had a couple of questions about what the discrete data points on there represent. Some of them I could figure them out, some I couldn't. Some of them have quite big error bars. A couple of them don't. So for some of them, the error bars don't seem to overlap the fitted curves.
Yeah.
I was wondering if you could just talk a little bit more about the inference that you're doing there, please.
Yeah, so the way that how we got this data, it's not that we just looked at the genealogical relationships between individuals, but we actually we counted all the possible paths, and we used Mark Abney's IDCoF for that, which was a month of a server, 50 CPUs to go over the data and really do this thing. And we got all the 9 coefficients of identity for individuals. And then we calculated the IBD, the expected IBD based on the tree. And what we have over here, so we have some points. So no, this one was a bit, the confidence interval is after the curves. I don't know how to explain this point, but this is basically what we see in the data. These are not really like classes of relatives per se. We just stratify the data every 5% over here. But when we analyze the data, we just took the raw. We didn't first stratify and feed the line. We took the raw data and then we feed these 3 curves. Okay? Yes.
In this graph, you then said something about dominance and about epistasis.
Yeah.
Don't you have to get other kinds of relatives to be able to do that? To estimate the dominance, you need SIB-SIB correlation, and epistasis, you need, because MZ twin correlation is going to include both of those as well. How do you separate them all out?
Yeah, so what we did, we look for dominancy, we look at parent-child correlation versus SIB-SIB correlation. I didn't show the data here in this slide, but we look at the correlation of parent versus child, and then SIB versus SIB. This is not confounded by epistasis at all. And we detected a small signal that could be attributed to dominancy, about 3%. And then we also basically what we did, we just took the data. Since we have also the identity coefficient number 7 that relates to dominancy, we could subtract it from the correlation that we see. And also we fit, of course, the monozygotic twins. We also fit it, and again, it's like this line was the best line that described the data. And I can also show you this graph later on that we have. So dominancy can only confound, basically can create epistasis, but you cannot eliminate epistasis. So even if we didn't control for dominancy in this graph, it should not confound it by epistasis, right? It was supposed to be the opposite.
So if you're correct, then you should be able to get the environmental contribution too, if the correlation between MZ twins is less than 100%, right?
The correlation between twins is less than 100% for sure, but this is not the common environment. It's not what's called the household environment. I think these are just random chances. People die from various reasons, and there is a lot of conflict, just noise that we have. So it's about 80% or 75% of the correlation, basically it's just noise, what we see over there in the variance of longevity. And it can be wars, it can be other things that affect the data. Thank you.